Thank you. So I'll give an update on the research grant that we obtained last year, and you'll hear an update on the work we have done on that research grant. So I'd like to thank Saikagaku Corporation and Kaken Pharmaceutical to sponsor this research grant. So innate immune memory is a concept that we know from innate immune cells. It tells us when a cell is exposed to a pro-inflammatory factor, like bacteria or alarmins that can signal through toll-like receptors, the cells undergoes epigenetic changes. And these epigenetic changes provides an immune memory to the cell. So the second time the cell is exposed to the same or a similar pro-inflammatory factor, the inflammatory response of the cells is modulated. It can be lower, then we call it induced tolerance, or it's higher, then we call it trained immunity. So the question is, do disc cells have an innate immune memory? Well, it would be beneficial because the intervertebral disc is an immune-privileged organ and as such does not have an immune system. And that means if there are bacteria in the disc, it cannot be combated by the immune system. So in fact, the immune cells has taken on some the disc cells have taken on some innate immune functions like phagocyting bacteria, as you can see on the right side, or the expression of toll-like receptors. So toll-like receptor signaling is very important in disc cells and in disc degeneration. So bacteria, like the Cutibacterium acnes, for example, signals through toll-like receptor 2 and 4. And alarmins that can occur from fragments of the extracellular matrix also signal through toll-like receptors like toll-like receptor 2. So our group and other groups have shown that in discs with modic changes, toll-like receptor signaling and again toll-like receptor 2 signaling is important and enhanced. And this is because we have, in some of those discs, we have more Cutibacterium acnes. We do have more of the extracellular matrix fragments, as Tamara Menges has shown on Tuesday. Tamara also showed yesterday that we have an enhanced toll-like receptor 2 signaling in the end plate of modic changes. And from previous work, we know that we have a higher inflammatory cytokine release from discs with modic changes. So overall, we have good evidence that there is an enhanced toll-like receptor 2 signaling in discs with modic changes. So the hypothesis of that project is that bacteria induce trained immunity in disc cells coming from modic levels. And the project consists of two goals. So the first goal is to identify bacteria that associate with pro-inflammatory epigenetic changes. And the second goal is to identify bacteria that can cause the observed epigenetic changes and then show that these epigenetic changes cause the trained immunity. So in this grant, we were focusing on the first goal. So in aim 1, we were looking at the disc microbiome with metagenomics. In aim 2, we were then looking at the epigenetic changes by investigating DNA methylation. And in aim 3, we were then correlating these two data sets. So in aim 1, we did the metagenomic analysis of 70 intervertebral discs taken from lumbar spinal fusion patients. Some had modic changes, others not. There was no difference in the degree of degeneration. Overall, we observed over 50 different bacterial genera. And when it comes down to the modic changes, we saw in the modic disc that we have more bacteria overall, but a lower diversity. So we have more, but of the same. So this study has now been published in JUR Spine. In aim 2, we looked at DNA methylation with reduced representation bisulfite sequencing on a subset of the same intervertebral discs. And what we found is overall a moderate differential methylation in the modic discs. And the top hits, the top genes that we identified have been already associated with rheumatic diseases before. So in aim 3, we were then correlating these two data sets, and this is still ongoing work. So the goal here is to identify which bacteria cause what epigenetic changes and to which genes do they associate with. So this is one type of analysis we have done. So we were looking which bacteria, for example, correlate with genes that come from the toll-like receptor 2 signaling pathway. So here we identified 3 different bacteria that significantly correlated with toll-like receptor 2 signaling genes. And these bacteria have previously already been identified or cultured from discs. So in conclusion, this is the first project the first report that links dysbiosis to molecular changes in discs. And this helps to determine what are commensals in the disc and what are actually pathogenic bacteria. And by that, we overcome also this so-called ascertainment bias by just focusing on Cutibacterium acnes. And the data that was generated within this grant has now been used for larger grant applications. Again, I want to thank the Saikagaku Corporation and the Kaken Pharmaceutical Company to sponsor this research grant. Thank you.